It's my pleasure to welcome our last speaker, Luk Vandenberghe. Luk is an associate professor of ophthalmology, and he's the associate director of the Ocular Genomics Institute. He also holds the Grousbeck Family Chair in Gene Therapy and is the director of the Grousbeck Gene Therapy Center at Mass Eye and Ear. He will be talking today about his efforts in the last months to develop a vaccine for COVID-19 and the current status of that. Thank you, Luk.
Thank you, Rachel, for the introduction, and thank you for the organizers to host me here. I will try to take control of the presentation here, and in the next 7 minutes, hope to walk you through a program really that is not much older than 5 months and initiated in my laboratory at Mass Eye and Ear as a small research project. And while I'll tell you about the progress that we've made, one of the messages I want to make clear here is that, as you've seen in some of these other presentations, is that through the urgency and obviously the significance of this crisis, what has become clear is that while our trade is both directly impacted, but also identified as highly relevant in terms of finding ways to continue care and possibly find a resolution out of this, that the community has really stepped up and come together. And this is something that I've witnessed in this program. And I want to make clear here that really at all levels, this organization, this department, as well as many other organizations that we partnered with, have stepped up to work towards a gene-based vaccine, which we call AAVCOVID. The partner organizations are all listed here, and we will come back to in a second as well when we give you more detail as to the program. Before I start, a few disclosures. This particular program has some IP related to it, and one of our partners, Novartis, I have consulted with in the past. Other disclosures are not relevant to this program. Jim did an excellent job at introducing to you COVID-19 and SARS-CoV-2, illustrated here in a cartoon. A lot of the focus that I will be talking about is these red mushroom-like feature on the virus called the spike protein. Why are we dealing with a pandemic? I think this is known to many here. We have a disease with a certain set of properties that is being able to roll like a tsunami across the globe, and only the mitigation approaches that we have been leveraging over the past couple of months have reduced the R. But in the longer term perspective, we all know that ultimately population immunity is going to have to be reached, and that population immunity can only be achieved with a vaccine without causing additional mortality and morbidity. So that is the goal of this program. And as you know, there's many different vaccine modalities historically that have been navigated. They can be classified in two major classes, the traditional vaccines as well as the genetic vaccines. And these gene-based vaccines are the vaccines that have reached the clinic and development the fastest, mainly because they are prone to be developed in a rapid pace because of the advent of molecular biology over the past 30 years. The vaccine I'll be talking about fits in that category. Most specifically, it is a recombinant viral vector platform that we are leveraging and that we're familiar with through our work, interestingly, that has a tangent, as you know, through the development of Luxturna with the field of ophthalmology. What we are encoding is the spike antigen, which is illustrated here. It's a rather complex protein, and it's that mushroom feature on top of the coronavirus, which is indeed where the name is derived from, because that also gives it the crown-like feature. This is a synopsis of the progress that we've made and the plans that are ahead for this particular program, which I'll spend a little bit of time on. Before going there, this program originated in my laboratory in May, and before finding the eye as really the center of the world, I have strayed previously in a variety of areas, including vaccine research. And really the reason why we picked up recognizing the urgency of what was going on in China and the Wuhan region we picked up an observation that we had made about 15 years before in a technology that we have developed, and this is what we started building on here, really first as a small research project that then was escalated to a clinical focused project towards the end of February, and we have not relented since. So very briefly, we're developing a gene-based vaccine with a spike antigen, which is going to be IM administered, and we believe that this can be done with a single dose prime, so a single administration. And the uniqueness about this particular program within the large space of other vaccine programs is that we use AAV as a delivery system, and I'll introduce that to you a little bit more. We decided early on that we couldn't wait and do business as usual, and decided at risk to start manufacturing this vaccine using industry standards at 2 different sites. So we selected 2 candidates very early on that then went into manufacturing around the middle of April. That manufacturing included 2 candidates with 2 suppliers. We have recently announced the parties that we are partnered with to do this. One party for one supply is Novartis of Exis. Another party is a European entity called ViralGen, and then other support entities. We aim to be in the clinic with a phase 1 study in the second half of 2020, and we are working daily to cut those timelines to get within that window there as soon as possible. In parallel, we're working with a variety of other groups to build a preclinical package that would include also challenge studies in non-human primates, as well as traditional toxicology studies. And for that, we partnered with the University of Pennsylvania, where I previously trained, specifically Jim Wilson. The phase 1 study itself is going to be done at Mass Eye and Ear with Mason Freeman as the PI. And in parallel to all of this activity, which is entirely done within the academic realm, we are having discussions with partners at scale to ultimately globally deploy this platform in the eventuality of success. With that, we have other parties that are compelled by our biology, including most recently the Bill and Melinda Gates Foundation. Very briefly, what is AAV? I think it's familiar to most of you. It is another viral strain that we have neutered in the lab over the past decades to make it into a replication defective particle that can encode a gene that upon in vivo transfer is expressed by the host. And in this case, the gene is the antigen, the SARS spike protein. On this platform, particularly in rare genetic disorders, there have been 3 drugs that have been developed. Many of you are familiar with Luxturna, but in fact, there's 2 other products that have been developed. So why do we propose AAV here as a vaccine modality, which is indeed different than many of the other vaccine modalities that are moving forward? There's a number of reasons which I won't spend too much time on, but partly we are leveraging the same advantages of any of the other gene-based vaccines, which are speed and the type of immune responses. We are building on the extensive safety records that AAV has with clinical experience over the past 25 years, 3 approved drugs, and indeed also a safety cushion in terms of dosing, because in vivo gene therapy applications, the dose can be as high as 100,000-fold higher compared to the doses that we are proposing here for vaccine application. Importantly, thinking about scalability and the ability to bring this to larger populations, this unique platform is established and has capacity in the industry with over 40 biotechs and large pharma players, as well as contract manufacturers being around. Biologically, AAV is known as a potent B-cell immunogen, and we believe that neutralizing antibodies will likely be one of the strongest correlates of protection for the COVID disease. And then on top of that, we built on a particular technology, which I won't have too much time to introduce to you, but very briefly, this technology is a unique AAV species that leads to transient expression because of its pro-inflammatory nature, has no pre-existing immunity in the human population, and has been shown previously by us to lead to high B and T cell responses in both mice and non-human primates in a vaccine context, both in HIV as well as in flu or in influenza. The lead candidates that we've moved forward are 2. One is a full-length spike protein Another protein is S1, which is a subcomponent of the spike protein, both of which have been engineered to optimize their antigenicity. These are the two candidates which are currently midstream in GMP production, and our current estimates are that we may have as many as 1 million doses from a first manufacturing run performed by our partners. The immunogenicity data, at least in mice for now, looks very promising, and this is a very dense graph that evaluates some of the first candidates that we have moved to in vivo trials. This is 1 month immunogenicity data, where we both look at binding antibodies to SARS, as well as live virus neutralization studies that we do in collaboration with BL3 and BL4 labs. These are exceptionally high binding antibody titers, as well as highly competitive titers for neutralizing antibodies. In fact, these are some of the highest titers that are currently being published in the literature by some of these competing platforms. That being said, we believe that in this particular scenario, we need as many shots at goal, and we don't see this at all as a competition between groups, but rather a competition against a single virus. Very briefly, we are proposing to the agency, this is the Food and Drug Administration, a rather large phase 1 study that evaluates both the dose ranging as well as different age cohorts, largely the ability to dose once, but also with some consideration to dose our vaccine twice. And this is a closing slide here. You obviously have heard many things about the broader vaccine landscape. There are lists with over 100 vaccines out there. Probably a short list of about 20 to 25 vaccines has reached a level of industrial uptake that are being produced and will likely make it to the clinic. Again, we believe that we need all of these shots at goal to fight an unknown enemies, as well as the particular condensation of the timeline in development. As you know, most vaccines are developed over years, if not decades. In this particular case, with a much shorter time frame, what we are really doing is working in the dark and making at-risk decisions to speed up this development. And that obviously is something where mistakes will be made, or vaccines that aren't as successful will progress and ultimately fail. So we need to be ready in our back pockets with a number of options. The biggest battle will likely be the battle around getting to that population immunity, which is getting to the level of doses to induce that. None of the vaccine modalities that are moving forward right now can reach the 4+ billion doses globally that we will ultimately need. There will also be differentiation along a number of axes, whether it's safety, efficacy, timelines, and scale, and certainly cost as well, all of which are being considered with very little data at this point, but hopefully we will get some triaging with more data over time. We do believe that AAV-COVID in these various areas, at least conceptually, is competitive, but obviously actual trials will teach us this. Again, and I want to close by where I started. This is a humble program, or at least it's a program that started humbly, but has been elevated very quickly to a level of reality to be relevant in this pandemic. And this could only be done by a consortium approach where people came together, including the leadership at Mass Pioneer, within the department, within our umbrella institution, Mass General Brigham. And I couldn't be more thankful to the folks on the ground, which are a large group of 50 plus folks that are either working at the bench or working in contracting, working in regulatory affairs, working on the manufacturing. So thank you for your attention. I'm looking forward to the panel discussion.